Good morning, everybody. As usual, we'll give it a minute for all of our attendees to jump in to the Zoom. Thanks for being with us today. We have a really great presentation coming up. This week, we have a few updates, and we want to get to them as quickly as possible. So we'll jump right into that. Talia will send out a link for the CME here shortly as well. So again, thanks for joining us. We'll start with our Chief of Staff, Dr. Megan Mahoney, has some updates on testing. Dr. Mahoney.
Yeah, thank you, Earl. Good morning, everyone. I just wanted to make sure that everyone was aware of an important update on the Santa Clara COVID testing guidelines. You can see them here. So effective immediately, essentially, any patient meeting the criteria below here who enters a Stanford medicine facility within Santa Clara County must be offered a test at the time of presentation. So these are patients that include those who are showing symptoms, those who have been exposed to someone with known COVID, those who have been sent to us by the county to get a COVID test. And then in addition to that, anyone who qualifies as an essential worker, and you can see the descriptions here. For criteria 1 through 3 or A through C, they need to receive that test within the next calendar day. But for the essential workers, they can be tested within 3 calendar days, and the testing frequency can be limited to every 14 days. And then the next slide. Thank you. And so this is for all established and non-established patients older than 6 months old. And once you place the order, a ticket will be generated so that the patient can schedule on MyHealth. Our testing sites are ready to schedule all of these patients within the indicated timeframes. And if the patient needs to, they can call the number listed here to schedule an appointment, and they can also be directed to the ED if they require immediate testing. I've also included some websites that are available to patients, and I just want to also send out a wellness message in general to stay safe and to stay masked, to come in for your N95 mask refitting, and to get your flu vaccine. Thank you, Earl. Thanks, everyone.
Dr. Moody, thank you so much. Appreciate those, and thanks for joining us this morning. We have also returning to join us as a panelist is Dr. Manisha Desai. Among many things, she's the director of the Quantitative Science Unit, and she has a few slides to share with us about some interesting research work that's going on. Dr. Desai.
Thank you, Errol. I want to thank you for allowing me to give all of you an update today on an important trial. And the reason I wanted to speak to all of you today is because this trial could potentially involve you and your future participation. So as Errol mentioned, I direct the Quantitative Sciences Unit, and we've been coordinating from a data perspective many of the Stanford initiated trials in COVID. So today I want to debrief you on a trial that we designed to consolidate much of that effort to create efficiencies and to get answers to our questions faster and with fewer resources. So much of the work I'll talk about today is joint work with other statistical scientists in the QSU together with our clinical partners who are on the front lines of the pandemic. And I'll start by just motivating this type of trial. And then I'll give brief details of the trial itself, and I'll end by letting you know how you can get involved if you're interested. So as many of you here know, Stanford has organized a strong response to address COVID. So to date, Stanford's been involved in about 40 trials, and that includes our participation in global efforts like the ACT and ACTIVE trials. It also includes Stanford investigators initiating their own trials. And in addition, the COVID Trials Research Unit, which you've heard described by Dr. Upi Singh in this forum, was established with support from leadership to support such trials as well as other efforts. So with all of this great effort comes additional problems that we have to address and we have to be aware of. So in a recent article in Stat News, Bob Califf, who we all heard from last week, he's the former commissioner of the FDA and current head of clinical policy and strategy at Verily, he said that the system for figuring out the best treatment options is off course. That with all of the very many trials being conducted, the studies are often too small to answer questions. And indeed, the authors of this article reported on an analysis that showed 39% of the trials were enrolling too few patients to yield definitive results. So the authors pointed to the UK-based master trial, recovery, which we've also heard about here in Grand Rounds, as a model to follow, because recovery consolidates and coordinates that effort instead of distributing it across multiple single trials. And of course, we have examples like that here in the US, like the active trial, for which Stanford is a participant. So Stanford has worked hard to solve this problem, not just by participating in those global efforts like ACT and ACTIVE, but also by coordinating all of this effort. So for example, the School of Medicine established a prioritization committee where a number of things are considered, including availability of resources, like our patients, which is our most precious resource, as well as the potential public health and scientific impact. So because my team and I serve as the data coordinating center for a lot of these trials, where we're involved in many of the activities that are listed here, we saw opportunities to gain even greater efficiency, specifically in the design of these trials. And our solution has been to have a shared infrastructure, which involves a number of components, the most important of which is this master trial that I'm going to speak about now. So our trial is called COPS, and that stands for COVID-19 Outpatient Pragmatic Platform Study. So it's a trial for the outpatient, because when we were designing the study, there was great need for consolidation in that population. It's adaptive because arms can be dropped or added. It's flexible. We allow multiple endpoints tailored to the drug. So it can either be a viral-based endpoint or a clinical-based endpoint. And it's a master, which means we allow multiple therapies to be studied. And we do this efficiently by having a shared control group. And then I just want to hone in on the pragmatic nature of the trial and the features that make it hugely advantageous. So one is that there is one single IND for the platform. So you don't need to pursue individual INDs for each drug. Those can just be cross-referenced if they exist. So the process is really streamlined. There's no head-to-head comparisons between agents, and that's appealing to pharmaceutical companies because they might like the shared resource aspect of a master, but not necessarily want to compete, be in competition with other drugs. We have a shared control group, which is a huge advantage. As I mentioned, we can finish faster with fewer resources. We have a shared database, which means it's quicker to launch. There's no need for the prioritization committee to approve each sub-protocol. They just need to approve the concept of the master. And then we go to the FDA each time we want to onboard a new drug. There's no need for coordination of separate teams in the COVID trials research unit. So here's how it works in practice. You start with randomization across the drugs that are currently on the table. As data come in, you assess whether that drug should be graduated or stopped for futility. If that's the case, randomization would be revised and enrollment would continue. And at any point throughout the study, a new treatment arm can be onboarded, which would also prompt revision of the randomization scheme. So there's a number of challenges, as you can imagine, to designing a study like this. The biggest, and I think it's something that Stanford excels at, is gaining consensus among partners. So you have to have investigators that come together, and the investigators need to come together with regulatory agencies and with pharma to agree on things like what's the endpoint that we're going to use to judge treatment strategies? How are we going to decide go, no-go decisions? And you can imagine the logistical issues that need to be worked out, like how do we maintain the blind with different looking placebos? What if the eligibility criteria for one drug is different than another? What if a site wants to partner with us, but they're only interested in trying out or assessing a subset of the drugs? And these are all issues that we've worked on and we've figured out. So if you're interested in learning more, if you're interested in possibly participating in COPS, please contact me. And I just want to give a shout out and acknowledge all of those who helped us get to this point, who helped us to get to launch. Some of those folks are listed here. There will be additional members of this team. So I'm looking forward to growing this team as we move to implementation, but I did want to thank everyone for getting us to this point. And Errol, thank you for letting me visit today to debrief all of you.
No, Dr. Desai, thank you for joining and doing this work and sharing it with us. So please keep us updated along the way. And again, thanks for being with us today. We'll quickly move over next to Dr. Brian Bowman, who's heading occupational health. Thanks, Dr. Bowman, for joining us.
Thank you, Errol. I just want to share a couple things this morning. First is the same employee testing chart that you've seen before, showing basically stable number of and small number of positives over the last several weeks. Of course, we check closely with any positive testing employee and do contact tracing where necessary. Nearly all of these infections, not all, but nearly all, are community acquired, and we haven't seen any significant spread within the organization recently. At the same time, I just want to bring folks' attention to, well, a lot of you have probably already heard about the outbreak at Brigham and Women's Hospital recently, and I just want to use that as a goad to us to continue our vigilance. Now, in press reports of that incident, it appears that there may have been lapses in PPE. And, you know, I'm preaching to the choir again, but we need to continue to be really vigilant and continue to encourage all of the employees and staff that we work with to be really vigilant about that. We're going to try to get, those of us who have some contacts at Brigham, we'll try to get more direct information and see what we can learn from that outbreak. But it just serves to remind us that although we see pretty stable data and we're doing really well, we've got to continue to be careful. The other thing I wanted to share today is just that as of tomorrow, we are launching our self-scheduling process for employees who wish to have a COVID test or who need to have a COVID test. But really either way, in parallel to the county order that Megan went over so nicely, we also are allowing any employee who wants COVID testing to simply sign up and have a test. The only restriction being that at this time, we're not allowing tests without any particular reason more often than every 2 weeks. So if anybody needs a test, wants a test, easy to get at this point. Any questions, just let me know, but it's available. And that's it for today, guys.
Dr. Bowman, that's great to hear. Again, thanks for joining and thank you for the updates. And for our last update, we'll jump in with Dr. Will Collins, Dr. Huja, who's been updating on a lot of the census numbers of COVID in-house, still with us on the panelist group to help answer questions. But Dr. Collins has been also helping gather the data this whole time and is going to be regularly helping us keep updated. Dr. Collins, thanks for joining.
Thanks, Errol. Thanks to Neera too for doing these updates, and I'm really excited to continue with them. So we can go to the next slide. So I'm going to update on our inpatient numbers, but just thought I'd quickly show a slide on sort of globally where we're at with COVID-19. I think a lot of this is known by folks, but this is showing active cases as of last night from the Johns Hopkins resource. And then unfortunately, we are now reaching some kind of grim milestones of over 1 million deaths globally and over 200,000 deaths in the US that we've passed in the last week or so. So next slide. I guess on the good news side, at least for the Bay Area, looking at the 7 Bay Area counties, this is both confirmed and suspected. The numbers still are trending down, and you can show the next slide. And this is breaking out all 7 counties. Again, you can see each county, generally speaking, the numbers are trending down. We'll go to the next slide. And when we look at our trends here at Stanford too, we see essentially the same thing where we had this peak at the end of July, and then we've still seen overall a good trend in our numbers. So we can go to the next slide. Oh, sorry, this didn't get updated, but as of this morning, we had, I believe, 4 PUI, 6 COVID patients, 1 in the ICU. I'm not sure whether or not that person was intubated. But looking at our data through September 27th, we've had 467 at Stanford Hospital, still around 20% or so that have needed ICU care. And our number of deaths is the same as last week, 31. We can go to the next slide. We remain having a slight predominance actually of females. Our age distribution is here. That hasn't changed all that much, but we certainly see patients across all age ranges who have been coming in. And then we can go to the next slide. And then the latest update from pediatric numbers, we've seen 45 patients now admitted. So we can go to the next slide. Just looking at the deaths again, as I said, there's no change in the numbers since last week. It's been slightly more males than females, around 60%, with the minimum, maximum age, and median age shown here. And then the next slide. And this is showing sort of our age distribution of deaths for all patients, and then for female and for male patients. Again, this is the same as it was in the last week. So going forward, next slide. So I just wanted to thank everyone, thank Mira, thank of course Isabel, who's helped with us, as well as Jake and Rudy, who help us gather these numbers every week. And I'll send it back to you, Earl.
Dr. Collins, thanks so much. Greatly appreciate you joining us today. I want to just briefly mention that we have next week's presentation is Dr. Shishianand, Dr. Parsonet, and Dr. Rath. We're talking about COVID-19 in dialysis patients, particularly in renal patients. So looking forward to that. So I'd like to just hop in now to our main presentation. Dr. Michael Snyder, he's the chair of the Department of Genetics and has been chair since 2009. He's also the director for the Center of Genomics and Personalized Medicine. And Dr. Schneider has done a lot of amazing work throughout his career. He's the first to perform large-scale functional genomics. Him and his team discovered a lot of different things that have helped medicine tremendously, such as that there was much more to the human genome, such as transcription and other things that were playing a role in regulatory information, characterizing high diversity of transcription factors and binding within and between species. Really, Dr. Schneider, the one thing I learned in learning more about you is that I could easily spend this whole hour talking about all the amazing work you've done throughout your career and are still working on now. I'll just say this, thank you for joining us today. Dr. Schneider also, a few years ago, authored a book called Genomics and Personalized Medicine, What Everyone Needs to Know. In that book includes chapters on the very thing he's talking about today, and the use of wearables, specifically in COVID-19, but really he talks about the use of wearables and really in big data and how we see the future of medicine. So ultimately, Dr. Schneider, what's so great about what you do is it's really the cutting edge of the future of medicine that we'll be all seeing. So you're playing a pivotal role in that. Thank you so much for joining us today. I'm going to go ahead and turn it over to you now. I'll stop sharing my slides so you can start sharing yours.
Great. Well, Errol, thanks very much for having me here. Switch over. And I have to say, I've given grand rounds before, and I think they've been reasonably well attended, but I can safely say I have never had 500 people attend one of my grand rounds before. So this is absolutely amazing. So I'm very delighted to be here and show you what we've been up to. This is all very, very recent work. Most has just been accepted for publication, but not yet out. So I'm going to start with the intro, picks up exactly where Will left off. This is a slide adapted from Rob Jackler. He sends around these weekly notices. I'm sure many of you get them about, tracking cases and deaths and stuff. And so you saw the first wave that came out in April and May, much stronger one in July. And although it's been going down, including the Bay Area, there are many areas of the country where this is actually going up very, very rapidly. So we're not even close to getting out of this pandemic. And so the way we're trying to test for COVID-19 is using PCR, as many of you know, at least for active infections. And this is normally slow. It takes a few days for a test. You can get it turned around in a day, but typically it's 2 to 3 days. And it's also fairly expensive. It depends on who you talk to, but it's $120 a test. And so what that means is that when you're trying to run millions of tests on millions of people on a very frequent basis, it's just not, quite frankly, scalable. I don't think this is the way to get out of this pandemic via testing. You're just not going to reach tens and tens of millions of people every day, all the time with this kind of technology. It's just too expensive and very difficult to implement. So we've been thinking about wearables, which is something we've been working on for some time, as a way to actually get out of this. So why wearables? Well, for one thing, they're worn by millions of people already. 20% of the US, over 50 million people, wear a smartwatch. One simple device can make hundreds of thousands of physiological measurements on you throughout the day. So in fact, you can get sampled at very high frequency, and they can track many different kinds of things. So the slide's a bit dated, but wearables today will track your resting heart rate variability, Many of them do respiration rate, blood oxygen, skin temperature, even blood pressure to some extent can be measured. Some of these are quite accurate. Some of them are not so accurate, but the deltas, the change in those can be quite powerful. And so we've been, testing out different devices, seeing how accurate they are, what they wear, and we've actually been using them in our research. And in fact, I'm a big believer in this technology. Many of you may know, I actually use 8 of these devices every day. Here are 4 smartwatches. I usually wear a ring, but I lost it recently. So anyway, I'm a big believer in these technologies. I think they're going to be quite powerful. Why are they powerful? Well, they're measuring you once again all day, and you can actually start seeing people's patterns. So this happens to be a few months of data from me. This is my circadian pattern. So I sleep at night, wake during the day, occasional nap, back to sleep at night. This is heart rate, which is normally low at night, high during day, back down. Skin temperature, which is different from oral temperature. For most people, high at night, drops in day, comes back. So that's me. We can measure these for lots of different people. We've now put these on thousands of people, and everybody has different measurements. So I have different resting heart rates at different times during the day, different skin temperature, so on and so forth. So we all have physiological patterns that differ from one person to the next. And that's very, very critical if you're going to try to detect disease. You need to find changes from people's healthy baseline. So the way we got into this, many of you may know if you've seen me talk before, is we're all about trying to bring big data and new technologies into better managing people's health. And so initially started with doing lots of detailed omics measurements on people, sequencing their genome, then lots of other detailed omics measurements, following people longitudinally. So there are 2 aspects of this. One is the big data, and the other is following people longitudinally. We tested this out on a group of 109 people. We've been profiling them for actually 7.5 years. And some point along the way, I think it was about 7.5 years ago, when wearables first started coming out, we realized that these were powerful devices, that we should be using them perhaps more than just for fitness trackers. We could actually use them as these physiological monitors I just mentioned. And so from the study, we actually, as I say, profiled people, and just from the first 3+ years of data, we actually came up with 49 major health discoveries. So almost half the people learned something important about their health. In some cases, a big deal, like we caught someone with early lymphoma, so on and so forth. And wearables did play a role in this. In 3 cases, it was no one technology found all these things. Different technologies teased out different things. We think they're all going to be powerful at different levels. But one person we caught with AFib from a wearable, another with sleep apnea, and actually a third person, which happens to be me, we actually figured out my Lyme disease from a smartwatch and a simple pulse ox. And so the backstory behind this was actually I was helping my brother put up fences in rural Massachusetts. Then 2 weeks later, flying to Norway through Frankfurt. And on that last flight, many of you may know, your blood oxygen drops on airline flights, and it varies with the pressure in the cabin, basically. And it turns out on this last flight, my blood oxygen dropped abnormally low. Normally it would drop like this on this sort of short flight, which is fairly well pressurized on that type of plane. But it dropped, instead of being at 96, it dropped to a median of 90. And when I landed, it did not return back to normal. So my blood oxygen was abnormally low. I was also looking at my smartwatch time. You can tell I measure myself a lot. And I discovered that my heart rate was actually running high as well, which surprised me. And it also did not return to normal after we landed. And then I later learned my skin temperature was elevated as well. And so in fact, I later got a fever. This is low resolution, but ultimately I went to a doctor after a few days. I warned him it might be Lyme because of the timing, and he drew blood. My monocytes were up, said, yep, I think you got a bacterial infection. He recommended penicillin, and I said, no, doxycycline. A little tense moment there. He gave in, and I took it for 2 weeks, but it did clear it up right away. When I got back, I was measured, and sure enough, I was Lyme positive by sera tests. I even had some antigens around. And it's very well controlled because I happened to have given blood 3 days before I left, and we looked at that sera and it was negative. So I sera-converted during this time. So that prompted us to look at all the data I had. I had 2 years of data at the time, and it turns out I was ill 4 times. One was the Lyme time, Twice were viral infections, we now know, this one and this one. And this is a big deal. The fourth one, this one here, it turns out I was ill as evidenced by CRP, but I didn't report having any symptoms. I only discovered it because I sample myself a lot. And it turns out that each of these times I had elevated heart rate and elevated skin temperature. So every time I was sick, I could see this retrospectively from a smartwatch by looking at heart rate and skin temperature. So that prompted us to write an algorithm that we call change of heart that actually looks at your normal baseline and then looks for a delta, change in your normal baseline, an elevation, if you will, in your resting heart rate at its earliest possible time in a statistically significant fashion. And what we discovered is that we could actually pick this up and in my 4 infections, every single time I could detect the illness prior to symptoms, typically about 30 hours or so, sometimes a little bit more. I'll come back to this later. And I had there were 3 other people in our cohort, one of whom got sick twice. It worked for them every single time as well, this algorithm. So we kept improving the algorithm. We were building infrastructure. This we published, you can see, a little over 3.5 years ago. And we kept improving this as we went along. And then, of course, COVID came along last March, or well, last January, depending what you want to call it. And with that, we obviously rapidly scaled up. We had an infectious disease study running, but we scaled it to ramp up quickly, had to get re-IRB approved. We have partnered with leading companies. Fitbit in particular has been very good to us. They give us 1,000 watches. I'll come back to that a little later. Garmin and others are all eager to work with us. And very, very quickly, we enrolled, now it's about 6,000, but we enrolled 5,000 people almost immediately. There were 32, if you will, what we call golden data sets, and I'll come back to that in a minute. So who do we get? Well, we mostly got, this is right after the enrollment, about 5,300 or so. We mostly got, white Americans. We did get some representation of other ethnic groups. We did get a number of people with other issues. I think they signed up because they were hoping to learn something. When we looked, they were supposed to have a wearable watch to join the study. Most did, not all. And Fitbit was by far the biggest device used by our participants out there. And that's not surprising. They are the biggest one. Apple Watch is number 2. Here's Garmin and so on. So these are the people who signed up, and these are the devices they had. Within this data set of 5,000, there were 32 that were very well annotated. We had other people who claimed they had COVID. These had mostly had confirmed diagnoses. Because they all had confirmed diagnoses. They had, for the most part, a diagnosis date and a symptom date. And that was very powerful because we wanted to see if we could tell differences in physiology from the smartwatch relative to when symptoms first appeared. So these are the 32 people. I'll come back to them in a minute. They're just aligned by their illness. This is that start of the black line here. And then this is when they claimed the illness, the symptoms disappeared. And I'll show you the app in a minute, but we could actually record daily symptoms for those who joined our study at the time, if they hadn't been sick yet. So anyway, this is the very first patient. So what we did was we started analyzing these with algorithms. And the first one we looked at was resting heart rate, elevation of resting heart rate, very similar to what we had done previously for Lyme and other things. And this is our very first patient. See this purple day here? That's when they were officially diagnosed with COVID. Day 47 is when their symptoms first appeared. But what you'll notice is that their elevation and resting heart rate started 9.5 days early. You can't miss a signal. It's very, very strong, very, very evident statistically in other ways. And I'll come back to this in a minute. We have an online detection method that we think can pick up COVID at its earliest times when you start getting ill. And again, well in advance of symptoms. So this person was probably running around for 9+ days, potentially spreading the disease without even knowing it because he didn't feel symptomatic till this day here and many days later. So we think this is going to be the power of these devices. So as I say, there are 32 people. It turns out for 26 of those 32, we could detect this elevation and resting heart rate. There were 6 people we missed. This is one example. Let's see, I got my bar in the way here. There's one example down here. I think that's this person. we missed some of the three of the 6 people had severe asthma or allergies, and we think maybe they're on medication, not quite sure, but their signals bounced around all over the place. We couldn't get a stable baseline on them, and we think that's why we couldn't find the elevation when they got COVID. And for 3 other people, we missed, and we're not sure why just yet. So it's not perfect. 6 out of 36, I think it's about 17%, maybe it's 15%, forgot the number, but we do miss with just a simple smartwatch, although I think if we add other parameters, maybe we'll do better. Anyway, it turns out for the most part, we could find this elevation of resting heart rate. We also wrote another algorithm I'll tell you about in a minute, where we look for, well, I'll tell you now, heart rate over steps. That actually can pick up, and we look for outliers in that. And we're doing this over this hourly interval because people change their patterns every hour over the day. And so we look for these deviations. And the bottom line is for 16 people, we had what we call a single bump where you'll get an elevation in resting heart rate or these other parameters. It turns out in most cases prior to symptoms. This is the symptom date. You can see this faint line here. And for 16 people, we pick it up nearly always before or at the time of symptoms. I'll come back to that in a minute. What was also interesting is that for 10 people, we had the same thing. We had a bump, if you will, an elevation around the symptoms, but they also had what we might call a pre-bump. They had another time of a stressor that elevated their heart rate. So it's possible that many of the people who got ill had other stressors associated with this that came up early. They're not necessarily COVID. I suspect they're not COVID, but that may, perhaps that makes them more susceptible. It's hard to say. But the bottom line is, in any case, 26 out of 32 people, we can tell when they're getting ill from a simple Fitbit smartwatch. Forgot to say, these are all the Fitbit ones. We do have others with Apple and things. They haven't been analyzed for technical reasons, but we'll happy to get into that in the questions. All right, so if we look at these various people, when can we pick up this elevation? Now let's just looking at the elevation of resting heart rate. Well, it turns out for the most part, the median is 4 days prior to symptom formation. So we can, again, find a median of 4 days before the symptoms come on. We actually detect this elevation in heart rate. And for some people, it's as many as 10 days. Some might be earlier, and that might be stressors, other stressors that bleed into this. But anyway, for these, it looks pretty clear as continuous signals. It also turns out to be 7 days prior to diagnosis. And it also turns out that we had 15 people who had non-COVID illnesses. Either we had measured them before COVID came along or they were diagnosed by other means. And it turns out they're positive as well in about 67% of the cases. And they will also show an elevated resting heart rate, in this case, a median of 2 days prior to symptom onset. And that's consistent with what we expected for our previous study. So it's not specific for COVID, it's detecting illness in general. We think that's still powerful in a pandemic. You want to know if somebody's getting sick, whether it's COVID or not. You don't want them running around spreading germs. And we think, again, the smartwatch can do this in many, but not all cases, not perfect. Okay, so this is what our summary. How much does the heart rate go up? Well, it goes up a median of about 7 beats per minute. It's a pretty wide range for some people, like this person here. It's about 2 beats a minute. That's plenty to pick up a signal because, again, you're measuring these over many, many hours. One person even jumped up, and it's not an artifact. They jumped up 62 beats per minute in resting heart rate over their normal baseline. It also turns out that people get COVID, they wind up walking less. We can do this retrospectively, and it turns out they actually sleep more if once they get a COVID infection, and we can show this again by a variety of means. We have correlated COVID onset with symptoms that people have reported, and in general, there's a pretty good correspondence. Here's a case where someone got ill, again, quite a few days, I think, prior to symptom onset. I think this was about 9 days, as I recall. They obviously start recording their symptoms at symptom on date. As it got more and more severe, you can see more and more symptoms have come in. Their heart rate is running high, and this other alarming system we have, heart rate over steps, can pick this up pretty strongly. In this case, it subsided. At least the illness seems to have subsided, came back, and sure enough, we can pick it up again with our algorithms. And so that was one person. Here's the case for another person, and so on. So in general, again, there's a pretty good correlation between picking this up with our algorithms on resting heart rate over steps, and symptom onset. So with this in mind, everything I showed you was retrospective, taking data, looking at it retrospectively. And what we want to do, and this is now phase 2 of the study, and this is going to roll out big time, I'm hoping next week, where what we want to do, instead of just detecting people after the fact, we want to tell you when we think you're getting sick because you've got an abnormal heart rate signal over your normal distribution. And so what we've done, I forgot to say, in this case is Meng Wang and Girish Boku, and I forgot to say there's a whole team of folks I'll acknowledge at the end who built a lot of what I showed you. A lot of this was originally built by Xiao Li in the lab. So anyway, we set up this alarming system. It's a 2-phase alarming system. The first alarm is set up around now the resting heart rate. That seems to work the best. We'll get, the first signal goes off in the first few hours. Now we've tuned it so it's 12 hours, but it can go off faster. And that's what we call a yellow alert. And then you get a fair number of those. So that kind of puts you on notice. And then we follow you. And if your second alarm goes off, that means something's really up, that you're way over your normal distribution. If it goes back to green, everything's fine. That's our 2-alarm system. And so everywhere we have a blue line is when the double alarm went off. Again, these are just some of the examples. This is an individual. This is their COVID illness, and the red is their symptoms. The purple is their diagnosis date. And again, this alarming, this is all retrospective, but the alarming will go off. We've tuned it on purpose in a way that it goes off about every 2 months for a normal healthy person. We have about 80 healthy people we've analyzed. So it goes off roughly every 2 months when you're healthy, and there may be things that trigger it. We'll come to that in a minute. But it then also goes off early enough. You don't have to wait, 3 days to see if you're ill, because that would be terrible. By then you'd be spreading it. So it'll go off, and we think we can pull this off in about 24 hours with about a 70%, 60 to 70% detection rate. So the point is that we think for 60 to 70% of time, we can tell you're getting ill within the first 24 hours. And again, it's generally well in advance of symptoms. Okay, so this is one example, an individual who, again, the alarm went off well in advance. Here's a second person. They had 3 alarms go up before their COVID, which is over here again, to go off early before this. We do see a lot of people do get bumps happening over the holidays. 4 out of 7 people where we had data there have what we call a holiday bump. It's over several days. This could be due to, stress from your in-laws, alcohol, travel, all these things we know induce heart rate. And can do it for extended periods. It could also be ill during that time. So there are other sources of these bumps, and you will have to contextualize the information. If you're watching a scary movie marathon that runs for 24 hours, maybe that'll keep your heart rate up as well. We know travel does this, by the way. Very, very long flights will keep your heart rate up for some time. If you go to Breckenridge, that will also put your heart rate up. So there are situations that will do it that are not associated with illness. But in some of these cases, we actually think people are getting ill, didn't necessarily report it. It doesn't just have to be COVID. Here's another case of a person who got ill non-COVID, and we can detect that as well. I'll show you this in a minute, but a lot of people, even more so with COVID, you'll actually see that there are abnormalities that persist after the original infection. Even healthy people will have these bumps. Again, the alarming setup to be every 2 months. Just because they're healthy doesn't mean they really are healthy. They may have gotten ill and be asymptomatic. We don't know. But these are abnormal, elevated resting heart rates that are running over reasonable periods of time. So again, we've set the alarming rate to go off roughly every 2 months. You can make it less sensitive, but then you'll be later in the infection. You can make it more sensitive, but then the alarm goes off more frequency. So we think we have a good count. So again, this is before people have COVID. The blue is how often these individuals will have an alarm go off. It is interesting, after COVID, the alarm just goes off a lot. We think there's a lot of complications and extended abnormalities that come after COVID infection. You'll see these post-illness bumps go for a while. They may not totally be post-ill. In general, I can tell you that the COVID peaks are generally higher. They're more severe, if you will, than other kinds of alarming bumps. So that is a bit of a clue. And the duration of a COVID alarming signal tends to be longer than that for both other illnesses and the bumps you see in healthy people as well. So again, we can pick up these bumps. We think we can generally pick them up pretty early. So then the question is, how do we get this out to millions of people? And that really is our goal. My goal is actually hit everyone on the planet. You may know that 60% of the planet has a smartphone. And if you compare those, even with a $20 smartwatch, you could do what I just described to you. You could actually tell people's elevation and resting heart rate all across the globe in real time. But you do need a computational structure to do it. And so there's a lot of challenges with this. Amir Bahmani and Aresh Alawi actually are the ones who built this beautiful system. I think it's amazing. I think we should actually probably use it at Stanford. It's highly secure. I'll get into that in a minute, but it pulls in data, stores it, distributes it, and we can do real-time analysis on it. Like we will, this is what we're trying to do. We're trying to capture everybody's wearable data, measure it in real time, and alarm them back that they're getting ill. If you think about it, that's a significant computational challenge, and we have to do it without breaking the bank. And so I think we've solved that problem. We've set up something that's scalable, it's interoperable, it can work through different kinds of data and different platforms, and it's highly secure. Stanford data security blessed us, which is a big deal. We're trying to be device agnostic. We want to pull in data from all these different devices. The heart rate data is pretty accurate, and we think we can work with that again and scale literally to tens of millions of people like instantly. And that's our goal. We're going to try and roll this out next week. So I'll show you how to sign up for a study in a minute. So we've set this up in a way where we've got apps set up on both Android and iPhone so that we can pull in your data, bring in surveys. We've set this up for COVID-19. We want it for any infectious disease. We're actually doing this for chronic fatigue and other things, other illnesses. So we actually have these studies set up right on your iPhone. We want you to enroll, join our study. Everything's super secure. You do say which device you're going to use. Right now, we're set up for Fitbit because it gives us the most high resolution data, and it's a pretty easy setup to do. To be able to do this in real time and follow millions of people, we had to set up and also keep it cheap. We actually set up using cloud computing. That's not surprising. We have sites all over the world that actually let us do this computing so that we can, again, compute in real time in areas of the world where the use is lower and keeps things cheaper so that we can save literally millions of dollars by actually doing this distributed computing. We have a great interface on the iPhone that can let you pull your data back so you can see all your wearable data. We've actually set it up so you can see your clinical measurements, your microbiome, your omics measurements, which interests us a lot as well. And you can dial up the resolution, whether you want to see this as daily, weekly, monthly, what have you, to see what, so you can follow your trends and your patterns. So the bottom line is what I've told you is that we've actually set up a method for detecting COVID-19. We can detect alterations in your physiology with a simple smartwatch that's associated with COVID-19. It's not specific for COVID-19. Again, it will detect other illnesses as well. We've built this online detection system. It's a little trickier, so it's not quite as sensitive, but it's doing reasonably well. I know when we add other kinds of data, we'll do even better, but this is what the data we've been able to get so far is the resting heart rate, steps, that kind of data. We've built a system to scale it to millions of people. I'd love to see Stanford use the platform as a general tool. I think we had to build it because we needed it quickly and take on this pandemic. I actually think this platform, because it's super secure, will be very valuable for both the hospital and research project at Stanford Medical School. It's actually, we're going to open source this. It is open source. And we're also going to open source our COVID-19 detection algorithms. We want you to sign up for our study. Here is, in fact, the website. Look at innovations.stanford.edu. Sign up now and join our study. We need to get your baseline data. And then as we roll out phase 2, which I'm hoping we're scaling it now a little bit. We're running at small numbers of people. Then we're about to literally roll out to millions of people. So we need to get your baseline data now so we can build a normal distribution so we can tell when you're getting sick. You'll see this elevation in heart rate, and we'll ping you back when we see this abnormal signal. If you're watching a scary movie or drinking a lot or what have you, just ignore it. But if you're sitting around listening to boring Mike Snyder and your heart rate's jumping up, well, then you probably are coming down with something, and maybe you want to get a follow-up test. So I've had an amazing team do this. As I say, the work was really led originally by Xiao Li and Jesse Dunn and several others. And then with the scale up, it's been incredible. We've got an amazing team of Xiao, Amir, Tejas, Ben, Alexander, Emily, Arash, Meng, Andy Brooks, Ahmed, and Garish are some of the principal leaders, but we have a whole back end building this infrastructure that we hope will, again, scale to the entire planet. That's our goal. So hopefully I've saved enough time for questions. Yep, it looks like I'm in perfect shape. So love to take any questions if you guys have any.
Dr. Snyder, thank you so much for that really interesting presentation. I think a lot of us found it really interesting just by noting the number of questions we already have. So thank you for saving time for questions because we have a lot of them. I'm going to jump right into it to get to as many as possible. And the first question, actually, I think I'm going to loop in a few of the questions because they have a similar theme. One person asked, were there a high rate of false positives? Another question was asked about any differences in gender, thinking about menopause might have temperature differences or perhaps another false positive here. And then Dr. Siegel asked, given what is known about the incubation period for COVID, this seems like picking up people before the event are being exposed. Again, I think hinting at, is there false positives? Can you comment along that theme?
Yeah, so I don't, what we can say is these are heart rate abnormalities, resting heart rate deviations, shall we say. Are they false positives? They're not COVID-19. That we know because some of them are before COVID-19 came on. Some of them people told us they were sick. They specifically said, I had a cold, I came down with this, and then a month later, I got COVID-19. So they're annotated. And in many cases, though, we don't know what they are. They're not annotated. And we've been following me since I've just had a period. I can actually tell, I've gone over my calendar for the last 4 years. I'd say about 3/4 of them, I know what these alarms are due to. They're off for me, they're often due to travel. If I travel to Australia, you're up in a plane flight where they drop the oxygen, the air pressure. And so what will happen is your heart rate goes up in response. I don't know if you know that, but that's what happens. Happens to everybody. And so if you're on a long enough flight, that will set off the alarm. Okay, so long flights will trigger, and so that's a major source for me. I did have a holiday bump that may or may not have been associated with an illness. I did have another time when I was truly ill. I reported being sick, and we went back and looked at my alarming signal, and sure enough, I had this elevated heart rate. So it was not a false positive in the sense that I was in fact ill, just not COVID-19 ill. So, and what's not clear out there is how often people are getting ill and are asymptomatic just in general, COVID-19 or not. And so that's why it's hard to answer what's a false positive and what's not. All I can tell you is that these are elevated signals. And again, some of them are going to be trivial. Some of them may turn out to be serious illnesses. We do want to, tune our algorithms better. We do know, for example, with menstrual cycle, people's heart rate changes about 2 beats a minute over that cycle, 2 to 3 beats a minute. We have not yet tuned our algorithms to take that into account. So I think that'll help us perfect things better. Around menopause, I don't have data on that yet. I mean, these are all great questions. Somebody asked me, well, what if you do prolonged marijuana use? I imagine that's also going to set off an alarm because your heart rate goes up, as some of you may know if you've ever had marijuana. Anyway, the point out of all this is that there will be other things that trigger the alarm. You will have to contextualize the information. If you can't contextualize it, well, maybe you do want to go in and get a PCR test.
Got it. Thanks.
Those are great questions, by the way.
Great response as well. Thank you so much. Dr. Schillinger asked an interesting question. How about beta blockers, heart rating limiting agents? Would they interfere with the sensitivity?
Yeah, I really do worry about medication. And that's why once people got symptoms, the heart rate stuff goes all over the map. Some people consistently go up and look like they're getting more and more ill, and other people are bouncing around like a yo-yo. So I think they may be on medications, you know, trying to treat their symptoms. And you mentioned beta blockers and things. You're outside my expertise there. Someone else who's on this call, I see some cardiologists already, are probably more qualified to answer that question than me. I can tell you though, the asthma people, they've been our biggest challenge in this detection, because I suspect they are on medication, and we're having this hard time, I mentioned, getting a normal distribution for those folks. So that's been a problem for, I hope that when we can bring in respiration rate, drop in blood oxygen, maybe that'll help tease out signals from that group of people. But right now we only have resting heart rate steps. We have a limited amount of information compared to what I hope to have soon.
Gotcha. Dr. Bonilla asked the question early on that I also wanted to ask you, but I don't know if you can answer. So if you decide to get one of those wearables, I think the question is, which one do you recommend? I know you want to stay agnostic, using the Fitbit right now, the Garmin you mentioned, a few in the app I saw, there was a few different other brands there.
Right. Well, we're trying to evaluate them all, and I'm conflicted. I have a company too that naturally I think is wonderful with this stuff. So we are using Fitbit now because they are the most dominant users out there. There are 30 million active users of Fitbit watches, and they are measuring a fair amount of items. You do need high enough resolution for us to do the alarming the way we want to do. We don't want daily report backs. We want hourly report backs, so we can tell at the earliest possible time when you're getting ill. And the Apple Watch is pulling in that information. We can't get access to it. The health kit is not high enough resolution for what we want to do, which is unfortunate, because I'd love to tap into the My Heart Count study going on here. A lot of smiles there. But we can't, because it's just too low resolution, unfortunately. So if we can get behind the wall at Apple and get access to that, I think we can set the alarming system up that we want to. But right now we can't, and so we can't alarm Apple users because, again, we don't have the high resolution data. I think if you can share it with us directly, we'll be able to do this. We're trying to set up a workaround to be able to do it, where people directly send us the info, we could actually alarm them. But if we have to go through Apple, it gets too low resolution. So, these are affecting our parameters. But ultimately, we hope right away, I'm pretty sure we'll get the Garmin and Samsung, and I hope Apple, but and Fitbit we're already on board with, and we'll bring in different kinds of information, again, at a resolution we think we can work with to set up early detection.
Sounds like they're all good instruments, not necessarily one that you would want to hype over the other one.
Yeah, well, the reason I'm wearing 4 is we're trying to figure out which is going to be the best for this sort of thing.
Got it. Dr. Harrington, I think you had a question for you.
Mike, that was really fantastic overview, and congratulations on all this work. Thanks for doing it. I want to get back to the beta blocking question, because it raises the bigger question of what I'll call the multidimensionality of the data, and something that was already asked, the false discovery rate. As there's more data available, at some point you would imagine you could have someone's genomic data, you could have their EHR data, imaging data. How do you start thinking about the false discovery rate? And because there's going to be relationships that you understand, and then there's going to be relationships that are there that you don't understand. And maybe we even want to pull Manisha and or Nigam into the conversation to give some thoughts.
Yeah, I'd love to hear their comments. Our view on this, it's all about well-annotated data. If we know what those bumps are due to, we can train on that. Now, I have a pretty good idea what a travel bump looks like now, what an airplane bump looks like, because I have enough data like that. We're getting a certain amount around COVID, but you raised some of the interesting issues. For example, is the bump different in diabetics versus not? And by getting that information, I forgot to say, but 78% of people agreed to share their EHR data with us. And that's fantastic. So hopefully we can bring in the beta blocker information and see how that correlates. And if we can also annotate it with when exactly people take their medicine, I hope to be able to get at some of the issues you just raised. So it's all a matter of having really well annotated data sets. Right now, we're well annotated around the COVID and the infectious disease. We're not so well annotated around the false alarms. And that's actually part of the app. That's now, as we roll out the beta study, or sorry, the big study, phase B, we want people to annotate. We're going to tell you when you have an alarm, and if it's a scary movie, you ping us back and say, scary movie, bozo, ignore it. But that'll help us understand what a scary movie signal looks like. I don't know if that'll affect respiration, for example, and some of the other things that would be powerful. It's not going to affect steps. So this is what we need, well-annotated data sets. It's a great question. I don't know if Nigam or Manisha want to weigh in on this.
So the way I've been thinking of this is an empirical characterization of the false discovery rate. And for example, right now, yes, there's elevated heart rate, and the label we have is COVID or Lyme disease, and so you can associate the elevated heart rate with that label. But that doesn't tell us that the association is specific to that label. There could be 50 other labels for which we would see the same trend, the slight elevation in the heart rate. And so how do you discriminate between the alternative labels? And so the experiment that can be done to empirically quantify that false discovery rate of that discrimination is to just assign, say, 50,000 random labels. And then if you're able to discriminate a large fraction of those random labels, then we know that the ascertainment of the COVID-19 classification is practically a fluke. And we'll need to do more of these empirical experiments as we deal with these ultra large feature spaces, and then convince ourselves that what things are really discernible and what are just discernible or appear to be discernible just because we haven't tested the alternatives.
Right. Yeah, that's a good way to put it. And we'll obviously do machine learning around these to figure out what features are specific for which situations.
Dr. Desai?
Yeah, I agree with the comments that are made. And I think this is more about sort of diagnostic testing and getting a really good characterization of sensitivity and specificity. And that's a lot of what Nigam was alluding to, and less so about false discovery rate. I'm less concerned about that, and more, I think the focus really has to be on characterizing sensitivity and specificity in the relevant population with all of those things that I think Nigam mentioned, things for which you want to make sure that the test has good specificity for.
Yeah, but the one thing I would like to add to this conversation is that for a lot of these things, I mean, it'll come up often, but we also have to pay attention to the cost. So there's certain things where it's okay to have a high false discovery rate because we don't want to miss the patient. HIV.
I think you mean sensitivity, like to have good sensitivity.
Yeah, but which means you will also have high false discovery rate or false positive rate, like HIV screening, being an example.
Right.
And then there are certain situations where if false positive is just nuisance and is going to lead to unnecessary investigation and is not worth it. And as a society, I don't think we have a clear guidance on how we decide which condition falls where. The extremes are clear. You don't want to miss somebody who's HIV positive and will tolerate a high false positive rate. But for other things, it's pretty fuzzy. And I think that's where some both theoretical and economic work needs to be done to figure out what should be our decision boundary and what is an acceptable trade-off amongst these things.
Agree with that, Nigam, yeah.
And to add to that, I think, these are very simple alarming systems. They are picking up something that's easy to follow up on. If you think about it, that's what a thermometer is. It's a pretty simple, and to be honest, it's a terrible measure of COVID illness, right? Plenty of people don't get elevated temperature. I don't know why we're running around using that, when I'm sure heart rate's much more sensitive than thermometer, but we use it anyway because it's easy, it's convenient, and it leads to a simple follow-up that you can actually act upon. And that's how I see these wearables as well. We picked a 2-month alarming, and to be honest, I want to let people set the dial. If you're in a healthcare situation, you may want to increase sensitivity versus being somewhere else, you may want to decrease sensitivity, right? So we want to actually let people control the dial as to how often they want the alarm to go off, depending on their personal preference and maybe their risk situation.
Dr. Schneider, and to the panelists, this is a great discussion. Do you mind if we go over a few minutes, maybe just 5 minutes after 9?
I'm cool.
Thanks. We'll surely won't get to all the questions, but we'll try to get to as many as we can. I have Dr. Rogers also on the panelist, and she had a comment or question as well. Dr. Rogers? I don't think we hear you yet. What I can do is come back to you in a second. I'll check with you again in a moment. But let me just pop up. There's a couple questions here I wanted to get to. Our surgical colleague, Dr. Daphne Lai, asked, would any of these changes in vital signs be for asymptomatic people? You actually just roughly touched on that, of course. Patients may not have fevers, but they may have heart rate elevations. Any data on that yet?
I don't. I'm trying to think. The problem is the devices we have now are mostly measuring heart rate and not skin temperature. Some of them do, but we don't have access to that data. So we'll know better about that from the, as we pull in other data from some of the other device folks and some of the newer watches as well. So we will have data on that. I do know for the asymptomatic case I had, there was no evidence of elevated temperature.
Gotcha.
There was elevated heart rate.
And you talk about, this is resting heart rate mostly, right? One question asked if the heart rate's elevated while running, is that taken into account, or is this mostly resting?
Yeah, we have done, that's where the heart rate over steps algorithm picks up that, and that has different sensitivity. We are playing with this. It relates to what we talked about earlier. We want to do machine learning and AI to figure out the best features. My intuition is that moment while people, either while they're walking or right after they finish walking, is a great time to be looking at people's heart rate and such, because a little bit of stress can really push things. Like, it's a stress echocardiogram, but for infectious disease, if you will. Sorry, Bob and everyone else I insulted with that sort of nomenclature, but you get the idea. If you add a little bit of stress, I think you'll actually get a more pronounced signal. And we just haven't learned yet the best way to pull that signal out. We're working on that. It's a great question.
Great. A couple questions here combining that talk about the population of COVID positive patients you are and have been looking at. What was the severity of the population? Were they hospitalized? And also, Sophia Yen asked, how are these patients checked for COVID? How frequently? Stuff like that.
Yeah, the ones that we dealt with in this first part, because we rolled this out right away, were PCR positive. The antibody tests weren't out yet. I think one of them had an antibody test, but they were basically PCR positive. Most were self-described as moderate. A few did wind up in the hospital, not necessarily right away down the road.
Gotcha. Christine Taylor's asking, can you please comment on the use of COVID tracker apps that have been very popular? So these are just apps without the trackers with them, especially the ones used in Britain.
Yeah, I mean, I don't have much experience. I haven't used them myself. I feel they're kind of late. If by the time you're tracking people when they have symptoms, you're already 4 days behind, but it's still probably better than nothing. I do think the concept is a good one. Obviously, there's the group out of Scripps that built, sort of followed flu infection and spreading by elevated heart rate in geographically mapped areas. So they could follow this retrospectively, how flu moved out. And in principle, you can do that with COVID as well. I actually like the idea where you might do this within a company or within Stanford, for example. If you know people's, exactly where they are and who they're interacting with and such, you actually could probably build a better contact tracer than exists now. And maybe you could even set it up. Certainly, some of them maybe you could set up before they actually show signs of illness, meaning we see the heart rate go up. And I think pulling data could be very interesting. If you think about it, imagine you're in a pod of 3 people, coworkers, whose heart rate all goes up, yet they're not symptomatic. You could actually get increased sensitivity. So I love the idea of pulling data to try and increase your signal. So, I'm not an epidemiologist, but that's probably about as close as I'll ever get to one.
Gotcha. I just want to mention Dr. Rogers' audio wasn't working, but she just wanted to make the point that she agreed with a lot of the comments made and to understand that many of the positives will be false positives, but in this situation, it's understandable to have a low specificity if you're trying to catch those positives. Dr. Rogers, thanks so much for joining and commenting, and your input's greatly appreciated always. A couple more questions and then we'll end. Again, thanks for going over a little bit. Dr. Schneider, you've done similar work I've read with the flu and trackers. Daniel Cap's asking, excellent work. Do you have any data or changes from influenza infections versus COVID-19?
No, all I know is that both of them elevate heart rate. The COVID one seems to have a longer asymptomatic period. We really need to pull in this other data, the respiratory rate, the blood oxygen, things like that, and see if we can tease out signals. I doubt that they'll be perfectly specific, but maybe we can see signals that will say, well, here's a 60% chance you're COVID versus, rhinovirus or something like that. I don't know yet. That's just conjecture.
Dr. Schneider, I'll end on this question, perhaps maybe the most important. How do you keep all your Fitbits charged? I see you guys wearables.
Every morning. It's amazing how much time I spend every day. I do more than Fitbits. I have exposometers. I have all kinds of devices. And as you might imagine, I devote a certain fraction of my day to charging my devices.
Well, Dr. Snyder, I want to thank you again for joining us. This is a clearly enlightening presentation. I still have 20 plus questions we haven't gotten to. If you're okay with it, we'll email you those questions. And if there's any you'd like to answer, we'll get them out to everybody right after this as well. Again, thank you for joining. As always, thank you for all our panelists for being with us today. And thanks for all the attendees for sticking along over after time here today. Hope everybody has a great week, and we'll see you next week. Thanks again.
Thank you. Bye, everybody.